The PKAN scientific community has faced serious challenges to advancing therapeutics. We're all aware of this. We've watched the literature since the gene discovery, since the new models started to come out. There have been models through the years, but each of them has limitations, including the models that we worked on with Jane Gitcher's group at UCSF. We've lacked a compelling mammalian model in which an abnormality could be specifically attributed to a defect in PANC2. And that's really important for this disease because we have an understanding based on our current state of knowledge, but I think our current state of knowledge is woefully inadequate to understand the role of PANC2 in cellular function. And so it was a determined effort on the part of our group to try and identify some kind of a phenotype in the knockout mouse that we had, and to try and develop that model. Without such a disease model, candidate therapeutics couldn't be evaluated with confidence. And this research resource really stood as a critical barrier to our field. And in similar ways, as you look at the other NBIA disorders, the state of the models, be they cellular models or organisms, are really central to the development of therapeutics for every single one. Without a model, also biomarker development was constrained, limiting our ability to identify sensitive non-clinical effects of new therapeutics. So we often focus on trying to identify the physical manifestations, the disease, if you will, manifestations in a clinical way, but sometimes the biochemical changes are just as powerful a readout or an outcome, and sometimes they're more powerful. So they precede the clinical changes that we see. This is true in human diseases as well. And if we're able to detect them, if we know what to look for and where to look, then we're able to find them. And so it really was the observations of Dr. Jeong and kind of bringing us full circle from the human disease, reminding us that this disease is exquisitely focal in the brain. If you look at the neuropathology outside of the globus pallidus, the brain is essentially normal. And she came to the group and actually helped us to develop this model that we had been working on at that stage for almost a decade. There was sort of an interesting coalescence or convergence of both the work in our laboratory and the opportunities that arose from work in the laboratory of Odie Seeboon in the Netherlands. She had identified a molecule that had the potential to replenish CoA and was downstream of the pantothenate kinase block in CoA synthesis. And so while we were developing a model that we were gaining confidence in, she and her group had come forward to the community with a candidate therapeutic and really important new insights into the biochemistry of this path. And so this has been a bench to bedside collaboration. You'll be hearing more throughout the morning. And what I want to emphasize here is that what the work started in the fruit fly, and we have always had the goal of getting it into humans. That's work that my group does only to a limited extent, and it's really been the fly work in Odie's group and the human work in Penny's group that has gotten us to a trial today. But in order to bridge those, we needed confidence in this molecule, and that confidence came in the mouse. The mouse developed now a decade ago, more than a decade ago, and the insights of a scientist who really has helped our group leap forward in our understanding of this disease. And so with this, I will turn the ownership of the screen over to Sue, and she can take it from here.
Thank you, Susan. And one note for myself, don't give the boss embarrassing picture ever in the lab. Okay, let me start sharing. Okay, hello everyone. It's great to meet you all here, even if it's online. And thank you, organizers, for making this happen. I'm mostly going to talk about our collaborative work that we published last year. It was published in IMO Molecular Medicine. However, I'm going to start how we started the story. So back in 2015, we published with Randy a postmortem PKAN brain paper that they show very striking similarity between the PKAN postmortem brain and the ischemic injuries. And this led us to a question whether there is a leaky blood-brain barrier in PKAN or PKAN models. At the time, we were using endothelial cells and the astrocyte co-culture from mouse brain. And after the flux analyses, Susan asked me whether there's something or anything we can do with this tiny monolayer of cells in an insert. I was like, okay, the only thing I can think about is qRT-PCR. So we decided to look at the CoA synthesis pathway enzymes, PPCDC, PPCS, and CoAZ. And interestingly, both PPCS and CoAZ was significantly downregulated, but they were so little, I couldn't even run statistical analysis. So we decided to look at another model using PANK2 knockout mouse brain. Excuse me. And when I trisected the mouse brain into GP-enriched area and SN-enriched area in cerebellum, it was uniquely downregulated in the GP-enriched area in the knockout mouse brain compared to the wild type. This was also confirmed using patient cells in vitro in culture that both CoAZ, fibroblasts and lymphoblasts showed decreased amount of CoAZ from the patient cells. And lastly, it was confirmed in the fresh uncultured blood cells from both mouse and human, shown here in the human and the mouse bottom here in the blue bars. This was particularly exciting because this opened the possibility for us to measure something from the shipped blood from patients and opened up the possibility for the remote trial that Penny will tell you later more in detail. This is directly involved in the coenzyme synthesis pathway, as we all know that PANK2 is involved in. But what about outside the coenzyme A synthesis pathway? So we looked into iron homeostasis. And this is a classical example of how iron gets regulated using IRP-IRE system. Excuse me. Transferrin receptor 1 transcript gets downgraded or degraded under high iron or iron sufficient condition because you don't need to bring in any more iron into the cell. And this is what we saw, that there was increased amount of total iron shown here on the right panel using ICP-MS from mouse brain, as well as decreased transferrin receptor 1 transcript, indicating that there is excess amount of iron in mouse brain. We also saw metabolic changes using patient cells, and they show decreased amount of respiration, and at the same time, increased ECAR, which indicate glycolytic shift in these cells, showing that, suggesting that there could be a problem with the mitochondrial function. One unexpected finding was change in dopaminergic pathway. That we saw a significant increase in both DRD1 and DRD2, which are dopamine receptor 1 and 2, maybe suggesting that there is a decrease in amount of ligand, which is dopamine, in the mouse brain, the PANK2 knockout mouse brain. If you are familiar with the last two NBIA meetings, you will remember that all the discussions went on for testing CoA synthesis pathway intermediates on PKAN models. So this is beautiful data from Odie's lab that when insect cells were treated with HOPAN, treating with 4-phosphopantethine, PPAN, was beneficial for cell survival, shown here in a red box. So we wanted to apply this idea to our models, both in vitro and in vivo, and cells from PKAN and the control primary fibroblasts and the pancreas knockout mouse were treated daily with PPAN. In cells case, it was 3 days, and then for mouse, it was orally for 2 weeks. And we saw what happened. So for 2 weeks of oral treatment, recovered Coasy expression in the in the knockout mouse GP completely in a dose-dependent manner. It also recovered transferrin receptor 1 transcript, as well as normalized the DRD1 transcript back to the normal level after 2 weeks of treatment. Not only a transcript, but in cells, it also restored some of the glycolytic shift changes in the PKAN fibroblast compared to the control, shown here in the dark blue bar. And lastly, in vivo, it also restored the PDH activity that was decreased in the knockout mouse to the normal level, as well as decreased complex 1 activity to the normal level by 2 weeks of oral treatment with PPAN. We have more data in the paper, but I'm going to stop here in terms of data, but also want to wrap up with what does this all mean, what we think what's going on as a current model. So this is the coenzyme synthesis pathway that we all know, that's starting from the vitamin B5 to all the way to the coenzyme A. And then when there's mutation in PANK2, we have PKAN. We reported various changes in 2 different models, both in mouse model and a cell culture model. The PDH activity has been gone down, maybe indicating the problem in the TCA cycle or the protein lipolation by mitochondrial fatty acid synthesis pathway. We saw decrease specifically in the mitochondrial kinase compared to the cytosol. We saw excess amount of iron accumulating in the mouse brain. We also saw complex 1 activity going down, suggesting problem with the OX-1 pathway in the mitochondria, and this is also known to be involved in the dopamine-induced homeostasis. And there is one interesting protein known to be involved in all of these pathways, and that is mitochondrial acyl carrier protein, shown here in the middle. And this protein is known to stay in biphasic status from apo to holoform and gets activated when the PPAN moiety get attached to the apoprotein become a holoprotein. And it's PPAN is coming from coenzyme A. So we think that this is a key protein connecting the CoA synthesis pathway problem to all the cellular problems we saw, and probably be playing a key role in a PKAN pathogenesis. Unfortunately, we could not detect a change in this holoprotein in our mouse model. We're still working on it with the OHSU protein core and try to detect this change in this key protein. But I believe that Odie will show you and tell you more about the changes in holoprotein in her talk. Okay, so this was a truly collaborative work. This is our NBIA cure team at OHSU, current and the former members. We're really a great team. Our collaborators in Netherlands, Odie and Marianne, we came a very long road together. Thank you so much. All the PPAN that we use was provided, synthesized and provided by Syncom in the Netherlands. NBIA, DA, Patty, and the team, we cannot thank you more. You guys rock. Excuse me, thanks to the OHSU cores and our department. And this is our funding source. Thank you very much. We're open for questions. And those of you who's observing runic calendar, happy Thanksgiving to you all.
Okay, thank you very much. The floor is now open for questions. Please use the Q&A button at the bottom of your screen or the chat for questions for either Drs. Hayflick or Dr. Jeong. Are there any questions? We have time for one or two. Well, I have a question actually for Dr. Jeong. Now, you see a downregulation of a handful of genes in your model and in the fibroblasts. Now, looking at those genes and the gene structure, do you see a common promoter element among those target genes that could be regulating them coordinately?
That's a very good question, but we have not looked at the promoter sequences. We were mostly focusing on the targets that are either in the CoA synthesis pathway or can be a downstream effector caused by the CoA problems. Some were unexpected, like DRD1, we totally didn't expect it to happen. But answer is no, but that's a good idea. Thank you.
And I have another question then. Have you measured CoA in your models to see if there is in fact a CoA deficiency?
Yes, we have. We've actually, in collaboration with you, we have measured the total CoA, and we also try to measure the free CoA, and we did not see the difference between our wild type and the control. Sorry, wild type and the control. Wild type and a knockout mouse model.
So how do you know there's a CoA deficiency?
That's a good question.
We don't. Well, we don't have direct evidence that there's a CoA deficiency, and it raises the question of whether we simply can't detect it because it's a compartmental, you know, it's a loss of maybe matrix CoA with a compensation, an increase in cytosolic so that the total is unchanged, yeah, we can't detect a difference in CoA, and we don't, I guess, we sort of build the story around a CoA deficiency, but I'm open to that not being true, that there isn't a CoA deficiency, that there's something about the biochemistry that we don't understand. Just to preempt the question, because many people ask it, can 4-phosphopantothene directly phosphopantothenylate ACP? And as best the literature knows today, the answer is no.
Well, that leads me to another question from the audience. And then in your ACP analysis, can you distinguish between APO and holo ACP?
So Odie has shown doublet, like a very tiny doublet in her Western gel, but it was in vitro overexpression system, and we could not detect that, at least to our power. So we're actually working with the proteomics core at OHSU to detect that holo and the APO form.
And along the same lines from the audience, this comes from Dr. Kurian. Do patients with PANK2 deficiency show any evidence of muscle complex 1 deficiency?
We have some reports, historical reports of people diagnosed ultimately with what we now know to be PKAN, but back in the olden days had muscle biopsies and mitochondrial complex activity assayed. And in 2 of them, I think there is a complex 1 deficiency, but it is not an isolated complex 1 deficiency. They also have changes in the other complexes. And so they were determined to be non-specific, but that may or may not be so. With a loss of complex 1 activity, you could argue that there could be some either downregulation or loss of function of some of the other complexes. So I think we don't really know the answer to that, and we have not prospectively collected muscle and looked. We haven't done it in the mice, and we haven't done it in the mice. So that might be worth doing.
Okay, well, thank you very much. We've run out of time. Additional questions, please. I'm asking the speakers to answer them online if you can, and we're going to move on to our next talk. Thank you very much.